Hey everyone. In this video, we're going to be talking about the primary structure of a protein. And so to get into that, we'll need to discuss peptide bonding, the N-terminus and C-terminus of a peptide, and then we'll get into primary structure. And the objective of this video is for you to be able to identify the primary structure of a given peptide. So let's talk about what peptides are. So amino acids can be thought of as a single link in the chain, and those amino acids can be linked together to form what are called peptides. They're connected by an amide bond, but when we're connecting two amino acids, we call that amide bond a peptide bond. Two amino acids stuck together is a dipeptide. Three is a tripeptide. Bunch of amino acids is a polypeptide. And then it's kind of a gray area, but usually like more than 50, we just call it a protein. But there is some overlap. Technically, any peptide is a protein, but we usually use the word protein for like really big ones. And the shorter ones we tend to call peptides, but there's no hard line. They're kind of synonymous terms. So let's look at forming a peptide from amino acids. So once again, the peptide bond is just an amide bond formed between the carboxylate group of one amino acid and the ammonium group of the next. So it's just a typical amidation reaction like we learned in chapter 14. We're going to be kicking out H2O here as water, and we're connecting the carbonyl straight to the nitrogen as an amide, highlighted in gray here, and the bond itself in red is the new amide or peptide bond. Since this is now a dipeptide made from two amino acids. Here's an example of a tripeptide. So you can take these three amino acids and we're going to connect them together. We're going to kick out our water here and here. So kick out a couple of those hydrogens and that oxygen as water. So we'll have plus two H2Os over here that got kicked out. And we are forming these peptide bonds. And so now we have a tripeptide made from three amino acids. Now, I want you to notice something about the structure here. Where you form the amide, it's always on the inside of the peptide chain. But on one side of the chain, you're always going to have this free NH3 group. This is the N-terminus, also abbreviated N prime. It's like the front of the peptide chain. On the other side, you have this carboxylic acid group that's free. We call this the C-terminus. It's basically the back side of the peptide, or the end. It's abbreviated as C prime. So you always have an N-terminus and a C-terminus, the start and the finish. And so here's just showing another example, right? Your N-terminal amino acid, because of the free nitrogen group, and the C-terminal, named because of the carbon on the end. So the N-terminus and the C-terminus. Now, when we start linking multiple peptides together, the order becomes important, right? In this case, we had leucine, then alanine, then tyrosine. But if we had had them in a different order, that would be a different molecule. And so the primary structure of a protein is the particular sequence of amino acids from N to C held together by peptide bonds. So let's take a look at this peptide here and let's try and come up with its primary sequence. And so we see that there's a peptide bond here and a peptide bond here. And so that's the dividing line between the first amino acid and the second amino acid and the third amino acid. Those are our 3 amino acids. Then we got to ask ourselves, what amino acid is it? And if we look at the side chains here, this is the alpha carbon right there. This is the alpha carbon, and that's the alpha carbon. And the side chain always sticks off the alpha carbon. So that's the first side chain, that's the second side chain, and that's the third side chain. And so we know that this side chain refers to valine. So this amino acid is valine. The middle one has just a single methyl group. That's alanine. And this last one is tyrosine. And if you don't have these memorized, that's totally fine, right? I'll provide you a chart with the 20 amino acids. So if you don't have it memorized, all you have to do is look at the side chain and go see which one it is. And that's how you would get the name. And we can use the 3-letter abbreviation. So from the N-terminus, we have valine connected to alanine, connected to tyrosine, and then we've got the C-terminus. So from the N-terminus to the C-terminus, this is the primary structure of this peptide. Valine, alanine, tyrosine. It's which amino acids it's made of from N to C. Now, all these examples I've been giving you are kind of in condensed form, but let's look at something in a skeleton form. So this is the way you'll probably see it on the exam. It's easier to draw peptides as skeletons using the software. And it's actually more accurate with the bond angles. And so if we look at this peptide here, we can quickly identify that that's the N-terminus, and over here is the C-terminus. Now, what we have is alternating alpha carbon, peptide bond, alpha carbon, peptide bond, alpha carbon, peptide bond, alpha carbon, peptide bond. Hopefully you're seeing the pattern by now. Alpha carbon, peptide bond, alpha carbon, peptide bond, alpha carbon, peptide bond, alpha carbon. Oh, and then we're at the end of the chain. And so this alternating pattern is accurate. When these amino acids bond, they actually flip-flop and alternate as they go down. And that has to do with just the shape of the molecule. The alpha carbon is tetrahedral. And so these make a 109-degree bend. And as they attach, they sort of flip-flop back and forth like your zigzag. And so drawing it this way with all the chains sticking down is not accurate. They don't actually all face the same way. They actually alternate. And what I've highlighted in blue here with all those alternating amides and alpha carbons, that's what we call the amide backbone. It's sort of like the spine, if you will. And the side chains are sticking out like ribs off the spine. And I'm going to erase here to make it easier to look at. In order to figure out the primary sequence, we need to look at the side chains. And so the side chains are always sticking off the alpha carbon. And so this is our first side chain. This is our second side chain, which looks weird because there's nothing there, but we'll get to that in a second. That's our third side chain, fourth side chain, fifth one, sixth one, seventh one, and eighth one. Now, we can identify the amino acids based on what those side chains are. If you had the chart of amino acids to compare, it would be a little easier for you. But I'll help out here. We know that this is lysine. Now, there's one amino acid where the side chain is just hydrogen. So when you draw it as a skeleton, the hydrogens are implied. So it just looks like there's nothing there. And it's kind of because there isn't anything there other than the hydrogens. And so that's actually glycine, the smallest amino acid. And this one is aspartic acid, ASP. This one is glutamic acid. Looks like we have another glutamic acid. Then we have a serine. And then we have a leucine. And then we have an alanine. And so our primary sequence from N to C would be lysine, glycine, aspartic acid, glutamic acid, another glutamic acid, serine, leucine, and alanine. And then we're at the C prime end. And so that is the primary sequence of this peptide. And that's your goal for this video, is to be able to do that on your own if you were given a chart of the 20 amino acids to reference. So thanks for watching, and I'll see you in the next video.